All right, so we're ready to get started. We're going to wrap up the last three slides that we had saved from chapter two, and then we're going to start into chapter three today. So we left off right before we started talking about ethics. And I always give a little bit of a disclaimer before this part of the lecture because ethics is one of those topics where you could take an entire course in ethics and still not give it enough of a coverage. So our three slides are not going to be the full picture here. So I'm just making you aware of that. Also, we're going to be looking at ethics, how they are specifically applying to psychology, and even more specifically, this is going to be biased towards human participants. There is a whole other set of or ethical principles that can be applied to non-human animals, but that is again beyond the scope of this course. I'm a lot more familiar with that side of things, but we'll do the human side of stuff because that's what most psychology is. So ethics, we're going to call those a set of general principles of how people should be educated, treated, and respected when they participate in any of our studies. So in general, it's basically a set of rules that tell us what's the best way to interact with people, how should we treat them, what rights do they have, how do we set up our experiments. So we'll see sort of the general rules of ethics divided up a bunch of different ways. One of the easiest ways to do it is the five basic principles, so A through E. Sometimes you'll see the same basic concepts condensed down into three principles. Both are the same basic ideas and pretty much inspire the same process, but we'll have slight differences based on not so much institution, but based on, say, country that it's being conducted in. So each country will have their set of rules. So the American Psychological Association, or APA, would be responsible for America. Canadian Psychological Association does Canada. They're going to be fairly similar to one another, but not necessarily identical. So let's talk very, very briefly about each of these principles. The first is the one of beneficence and non-malfeasance. And that's a fancy way of saying basically treat people in a way that benefits them, don't cause undue harm, and make sure that your research is going to be beneficial to society if there isn't a way to conduct it without causing some kind of discomfort. So when it comes to getting research approved, you would have to be able to say that if this is something that would make people uncomfortable or would even cause them harm, the benefits of conducting this research, the stuff that we might find, has to be much more valuable. It has to have more weight to it than that inconvenience. And so we have ethics review boards or internal review boards that would evaluate whether a study meets these principles. So there's a neutral third party at every university, every institution that conducts research would decide if your proposed project actually fits all of these principles. So just keeping that in the back of our mind. Our second principle is one of fidelity and responsibility, which is basically saying that you want to be trustworthy and behave responsibly. Don't lie to people, don't mislead them. Um, yeah, be a good person effectively. Principle C is about integrity. And so this is trying to be as accurate, truthful, and honest in your work as possible. So be a good scientist. Don't misrepresent your data. Don't manufacture data. Use good practices. So those are a little bit more on the abstract side of things. But next we get into some of the really, really important ones. So principle B is justice. This is one that says that we have to treat all groups of people fairly. So make sure that when you're selecting samples, that you select equally from the population that will benefit from the research. Don't bias your sample to those who are easy to pressure to come in and participate. Make sure that everyone is equally represented who could benefit from it. And we'll actually talk about an example where that wasn't done, which actually led to a lot of these principles existing today. But we want to finish what else justice encompasses. That's making sure that uh, we are aware of biases and do what we can to avoid any bias in our research. So, if you've talked about ethics before, you might have heard some of the big examples in psychology of things that pushed ethics to be what they are today. Things like the Stanford prison experiment or the Tuskegee syphilis study. There are so many different examples that we could point to because when psychology was just coming into its own as a field, we didn't have sort of larger governing boards that determined what was ethical and not ethical. So people could kind of do whatever experiments they wanted to, and sometimes they did ones that they probably shouldn't have. So, as relating to this principle of justice, I draw attention to the Tuskegee syphilis study because it directly violates and probably informed the creation of this policy because of that choosing or selecting your sample equally from a population. If you're not familiar with the study, that's basically when researchers wanted to understand how syphilis progressed and how they might treat it. And they did this by specifically targeting lower income, mostly Black neighborhoods and convincing them to participate in the study. We'll talk about coercion as a thing that's problematic, but usually it would be they'd offer them large sums of money to participate. They would get them to sign forms that they might not even be able to read. And then once they were in the study, they were the only people in the study. So even though everyone could benefit from finding a treatment for syphilis, they only targeted this marginalized group. And to make it even worse, this is more than what we need, but it's interesting. They once they even found a treatment for syphilis, they found that you could treat it with a dose of penicillin, I believe. They didn't give people that treatment because they wanted to see how it progressed. So that's one of those studies that I teach a methods in psychology section, and we have entire chapters on ethics. So it's one of those that we can point to and say, here's what has been done. Here's where all of these different principles of ethics have been violated in the past. This is why they exist moving forward. So that's one of those studies that was done in the past and helped push the field to sort of revolutionize how they approach these studies and leads us to having all of these principles today. And so the last of our principles, the respect for people's rights and dignity, kind of ties into stuff we've talked about there. The idea of people having autonomy, respecting their rights, and focusing in on informed consent, having them know what they're agreeing to when they participate in a study, and not coercing them into participating in that study. So these are really important things to make sure that the people who are in that study know the risk and they've made an informed decision to participate anyways. Now, we don't talk too much about it, but all of these principles are sort of put into place, and there's documents that dictate exactly what can and cannot count under each of these. There's massive documents that you can read if you do this kind of research, but typically there's sort of standards of what is approvable or not approvable depending on where you're doing research. And those internal review boards will take a look at any potential studies that you'd like to do and tell you if they approve it or not. And this is usually a pretty long process. Um, Again, I've done similar, but for non-human animals, where you basically write up everything, you list all of the potential problems, any way that you might cause harm, any potential risks, anything that could even be uncomfortable. All of those things need to be listed. And then you also have to include the potential benefits. What could you learn from this study? What are the benefits? What could we be working towards? And then the committee decides if the risks are worth that potential outcome. So that's the very basics of how that process works. I do want to take a moment and talk about some special ethical considerations. So we've mentioned this idea of informed consent a couple of times, and that's one of those big important things when it comes to ethics, especially in psychology. And with informed consent, we're basically making people, in order for people to agree to participate in the study, they have to know what the risks and benefits are of participating, and they have to understand and grasp those sort of pros and cons so they have all the information that they need to make an educated decision about whether they participate or not. So that's informed consent. But as you might anticipate just from that definition, there are some groups that can't give informed consent. So, for example, a lot of psychologists used to do, say, developmental psychology. Maybe they want to look at children. Children may not have the mental capacity to understand consequences and risks. So, what we'd have to do instead is have special rules and guidelines for how you can work with a group that can't make the decision for themselves. So, with children, you have to get informed consent from their parent or guardian. And then they would have to agree to participate sort of on their child's behalf. This also applies to other groups, maybe somebody who has a mental disorder that prevents them from thinking clearly or making decisions. We would then have to get that special permission from their guardian or parent, someone who can decide on their behalf and keep their best interests in mind. This is where we can also think about things like coercion, where people don't, or they have the right not to be pressured to participate in an experiment. So this is another thing that came out of my methods class, but I think to mention it because it's interesting and hadn't occurred to me previously. But there are certain structures, certain setups where people can feel pressured to participate in a study. Being offered large sums of money to participate in a study is one thing. That's actually one of the reasons why you're not allowed to be paid to donate blood in Canada. They don't want to coerce people who are, say, not as well off to have to go and give bodily fluids to survive. That's another of those differences between the US and Canada in terms of ethics rules. But you have to think of things like, say, you want to study how educational programs work in prison systems. Prisoners in the environment that they're in might feel pressured to participate in a study depending on how that study is presented. If a guard asks them if they want to participate, they might assume that they have to participate. They might assume that there are negative consequences to saying no. And so they'll participate not because they want to, but because they think they have to. So whenever you're dealing with a group that might feel undue pressure to participate, you have to take those extra steps to make sure that you counteract any coercion that might be existing. So lots of things to keep in mind there. And for our last point here, if you've thought this through, this concept of informed consent can actually be problematic for some of the ideas that we've talked about earlier. Specifically, if you tell people exactly what your experiment's about and how you're going to do it, doesn't that bias their behavior? We talked about how subjects can bias their responses. They can behave in a way that they think will support your hypothesis. Or maybe they're jerks and they're going to go in the opposite direction. But in either case, you're not getting an accurate measure of behavior. So that's problematic. So we need informed consent, but informed consent can also change their responses. And so what we can do is find sort of a happy medium with the use of approved deception. So if a study, in whatever way, doesn't reveal the entire true nature of the study until after they've completed their participation, we would call that deception. So this could be as extreme as you tell them that they're doing one thing while in fact you're studying something else that's happening in the background. But from my experience, assumption tends to happen on a much smaller scale. As I said, I don't do much work with humans, but I have helped someone set up a human study. And we had humans listen to Black Cap Chickadee songs. Surprise, surprise. But we didn't tell them that's what they were listening to. So our informed consent at the beginning basically said, you're going to listen to some sounds. And we're going to get you to watch or to listen to those sounds on headphones. And you're going to decide if you belong in this group or that group. You're going to click on a screen to indicate what group you think it belongs to, and then we'll give you feedback. The potential risks associated are, strain from hearing sounds, eye strain from staring at screens, wrist strain from using a mouse, the standard things that you have to itemize for experiments for ethical disclosure. So we disclosed all the potential risks, we just didn't give them the information that was critical to the study. And then at the end, we did something called a debriefing. And you have to do a debriefing if there's any form of deception. So every study is almost always going to have a debriefing. It's good practice to do, even if you haven't deceived anyone. And that's when you tell them what was actually going on. So at this debriefing, we'd say the sounds you were hearing were black cap chickadee songs. And we had two different populations. Some were from Ontario and some were from BC. And we were seeing if you can tell the difference based on their sound. And that would be in our just sort of our, I've already dropped the word, debriefing. There we go. I'm having a bad day for dropping words today. But that would be in the briefing at the end. If you had a much larger deception, had to lie about something or mislead someone, you'd have to make it clear what you did and why, as well as disclose any potential risks that you hadn't mentioned initially. And any form of deception has to be approved by that ethical board beforehand in order for it to go through. And again, so there are situations where larger deceptions can be approved if the benefits outweigh any potential risks. So it's always that weighing of pros and cons. Is it worth any discomfort to get that information? And the researchers don't get to make that call; those neutral boards do. But that's, again, a very, very simplified, condensed version of ethics as they apply to humans in psychology. So that is the end of chapter 2. And then we can start on chapter 3. Okay. And so before starting this chapter, I like to just give people a little bit of an idea about um, how we're moving through topics, how the topics connect, how we get here, and what we're trying to get out of this. So at the beginning of the semester, we started talking about psychology in general. What are some useful terms to know? What are some good ideas and concepts to know? What history is relevant going forward? Giving ourselves a nice baseline. And then we moved into talking about science in general and how to conduct good science and ways to think about science. So again, establishing that baseline. So this chapter, we're still giving ourselves a baseline, but now we're starting to give sort of more specific information. So we're going to get into the biological basis of behavior. Basically, we're going to look at the simplest units of the human body that contribute to our behavior. And so this is where we start getting into what people tend to think of as the real content. So it's going to be a different type of information versus what we've seen in previous chapters. So it's a less about theories and concepts and a little bit more about learning how things happen. So we're going to see this shift in how we talk about stuff. But this is actually one of my favorite chapters, and it's interesting because it gives us the information that we need to then build on it and understand how things happen in the future. So if you've taken biology, even high school level biology, some of this should be quite a bit of review. We're going to add some extra info. You'll find that we gloss over some stuff because it isn't as relevant. But we're going to focus in on what matters to us. And I always tell people that I take this chapter fairly slowly because I have no way of knowing how much or little background people have at the beginning. I might have a class who have all just finished taking cell bio, and this is exactly what they know already, and they know more than me. Or I might have a class that never took bio and has no idea what a cell is. So I move at sort of a mid rate, and I like to use actually some of the psychological principles we talk about when we get into learning, where I repeat things and I show them from different angles and I take my time. So if we finish where I intend to finish today, we're going to have explained a bunch of concepts and then we're going to stop before we put those concepts into action because I want to start the next lecture reviewing those concepts again, just giving us that extra chance for stuff to sink in. Because I do know that this chapter tends to stump some people. So just a little bit of an explanation as to why I present stuff the way that I do. All right, so with all of that out of the way, we're going to talk about neurons. So neurons are single cells that make up our nervous system. So these are cells that convey sensory information from our body and the environment into our brains. They carry out operations within the body. They can send instructions from the brain outward to muscles and organs. They have all sorts of different functions. So neurons are kind of the smallest unit that we care about when it comes to behavior. So these are the individual cells. And so we're going to start by actually looking at a generic uh, neuron here. So we will briefly point out different kinds of neurons and different shapes of neurons, but this is going to be our prototype image. This is our baseline to compare neurons against. And again, this might all be review for you. But uh, the first thing we want to point out is in the middle there, we have our cell body. So that's the part of the cell that is basically like a generic any other cell in the body. So it has the nucleus where all of our genetic material is stored. It has all of our organelles. So that's where the mitochondria are doing those powerhouse things. It's where the ribosomes are making proteins. That's where all of that cell stuff is happening. And because this is a physics class, we leave it there. We point out that it has a nucleus and cell stuff, and then we move on. But our cell body, also sometimes called the soma, is the part of the cell that's the main body of it. That's where all the stuff is happening that keeps it running as a cell. And we kind of ignore it from here on out. So next, the stuff that is more specialized for this to be a neuron, we can look at the dendrites. And these are these branching protrusions that are sticking out from our cell body. And you can think of dendrites as antennas that are bringing in or receiving signals from elsewhere. It might be bringing in sensory information from sensory receptors. Say they're taking in tactile information from our skin or taste information from our tongue. So they could be inputting information that way. Or they could be receiving signals from neighboring neurons. Maybe they're getting a signal that they have to pass along from the neuron ahead of them. So those dendrites are where we bring in information. Next, there's actually two things that are on our diagram that we're going to skip over until later. The first is the axon hillock, and the second is the myelin sheath. We're not going to talk about the myelin sheath until we start talking about actually transmitting information. So that'll become relevant later. And the axon hillock is where the cell body sort of connects the line between the myelin sheath and not myelin sheath. So those aren't relevant quite yet. So we can ignore those for now. But what we do care about is that whole sticking out protrusion that's labeled as an axon. So the myelin sheath is wrapped around parts of the axon, but the axon itself is a projection from that cell body, and it's pointing off and it's sort of reaching out towards other things. So the axon is going to allow the transmission of a signal that we'll discuss from the dendrites through the cell body and then along the axon in the direction of those red arrows. And then at the end of our axon, where it might be connecting to a neighboring cell, or in this case, a neighboring muscle, we have the axon terminals. And you'll see these called a bunch of different things. Just terminals, axon terminals. I've seen them called axon buds or terminal buds. All of them are referring to the same thing. And they're basically at the end of the axon, the cell is going to branch out a little bit and have little balls of increased surface area that allows them to transmit the signal to the next thing in the chain more easily. So we'll talk about that transmission process later on. But for now, we have the basic anatomy of a cell so we can orient ourselves as we start talking about what happens next. So I've already kind of hinted that there are different kinds of neurons because we talked about three different functions that neurons might have. So these types of neurons and what they do are going to map nicely onto that description that we had at the beginning of what a neuron is. So the first of our types of neurons is a motor neuron. So a motor neuron carries commands from our central nervous system, so usually from the brain via the spinal cord, out to the muscles and organs in the body. So motor neurons are that outputting of signal away from the central nervous system to do some kind of action. We have sensory neurons, and those are the ones that go from the sensory receptors inward. So they go from the outside into the central nervous system. Again, they're usually going to route through the spinal cord and then into the brain. And then the interneurons are the ones that exist between motor and sensory neurons. So interneurons connect one neuron to another in some part of the brain or spinal cord. And you'll find that we actually keep our definition of interneurons fairly vague here. And that's because they serve lots of different roles and take on lots of different shapes, and they're a lot more complicated than we can afford in this class. All right, so the next slide here, this is one of those for your information kinds of slides. This isn't something I'm going to test you on explicitly. This is just meant to give you an idea of the scope and different types of neurons that can exist. So I give that disclaimer so you don't panic looking at neurons. So on the far right-hand side, we have the example neuron that we've been looking at already. And if we were going to be precise about it, we could say that it is a multipolar motor neuron. And we might have figured out the motor neuron part because it's connecting to muscle fibers. So it must be a muscle or a motor neuron because it's sending a signal to the muscles. But that multipolar part of it is actually from the projections off of the cell body. This neuron here has lots of branching dendrites that stick off of the cell body there, so we call it multipolar. We can have a weird situation here at the very top, and that is going to be a unipolar sensory neuron. And so we might, again, figure out that it's a sensory neuron because it's pulling in information from the skin, from those receptors in the skin. So the dendrites are bringing in information from those sensory receptors. But you might notice that our dendrites here connect almost directly to the axon. The cell body is off to one side. So there's only one projection from the cell body and it becomes both the dendrites and the axon. And that's a special thing that has evolved in order to help speed up transmission. So because that signal coming in from the dendrites and going out the axon doesn't have to cross the cell body, we're saving time and energy. So this is a pretty standard orientation for sensory neurons, especially those that have to travel long distances or when time is of the essence. The other kind of sensory neuron that we will also see are these bipolar sensory neurons. And they just have one branch of dendrites and then the axon. So bipolar meaning there are two projections overall. And then we have, last but not least, our interneurons. And the only thing that they've labeled is the nucleus because the interneurons are those oddballs that kind of have a weird shape. They don't look like all the other kinds of neurons, and their function is that in-between thing. So that's kind of the perfect illustration about how much detail we're going to go into with the interneurons. But yeah, so that's the visual establishment. Now we're going to get back into a little bit of biology. And again, this might be review, but I'm going to explain it as though you haven't heard this before. So you might recall that our cell membrane is made up of what's called a phospholipid bilayer. And so we're going to break down why it's called that and how that specific orientation benefits us for the stuff that comes next. So first, we're going to look at what they mean by a phospholipid. So each of these little red circles with the two yellow tails on it, that would be a phospholipid. So we have a phosphate head, that's the red ball on the end, and then we have two lipid tails that stick off at the end. So when we're talking about sort of the orientation of these molecules, the qualities of those parts determine how they like to orient. So we can say that our phosphate head is actually hydrophilic. And if we look at the root of that, hydro meaning water, philic meaning loving, So it is a water-loving molecule. That means that the phosphate head doesn't mind facing outward into the cell or extracellular fluid, all of the fluid that's either on the outside of the cell, or it doesn't mind being on the inside of the cell facing into the intracellular fluid. So intracellular meaning it's on the inside of the cell, extracellular meaning it's on the external part of the cell. And so they'll orient so that it gets to hang out with water molecules because it has the right charge and the right shape to be okay with that. Our tails are made of lipids, and those fatty lipids don't like water. They're hydrophobic. They are afraid of water. And so they're going to orient away from those cellular fluids. They don't want to be on the outside of the cell, and they don't want to be on the inside of the cell. So they're going to organize so that all the tails get to hang out together, and they get to shelter each other from being exposed to any water that's in those fluids. So that's how we end up with this layer. So a bilayer, meaning there's two layers of them. So the phosphate heads are on the outside and on the inside where they get to touch water and they're happy. And the hydrophobic tails stay on the inside and mash together in two layers so that they're on the inside away from water and they're all happy. And that's how we get this setup. Now, this is really important because in order for a molecule to pass through this kind of an orientation, it would have to be cool hanging out with hydrophilic heads. It would have to have the charges that allow it to be around water, but then it would also need to not have charges so that it could hang out with the tails that are hydrophobic. And you can't have both of those things in the same molecule to pass through both seamlessly. So this setup here, this bilayer, ends up keeping things from traveling across the cell membrane. So if something is outside the cell, it's stuck outside the cell. And if it's inside the cell, it's stuck inside the cell unless we move it or unless we permit it to move by some other means. So that's our basic setup here where we're trapping some things outside and some things inside, and now we can talk about how we allow things to move across that theoretically impassable barrier. So the first of these important things to talk about are these green protein channels. And again, we can call them lots of different things. You can call them protein channels, you can call them ion channels. Usually we're going to be calling them by the name of the molecule that they move. So we might talk about a sodium ion channel. That is a channel made out of protein that moves sodium ions. So when we're talking about most of these things that embed themselves in that phospholipid bilayer, they're usually proteins, which is just a molecule that's manufactured and has a shape that lets it do the function we care about. So here, these protein channels are going to embed themselves in that cell membrane. They're designed to sort of fit in with the phospholipids. They're okay with that. And when they're open, they can allow different ions to move from the outside to the inside or the inside to the outside in a controlled manner. So our sodium ion channels are going to only allow sodium ions to move, and they're going to move in a particular direction. We can have different kinds of these channels, and they're going to be specific to different kinds of molecules. And they're not always open. They'll typically sit closed, which means that they are gated. So if you trigger them properly, they can open up and allow stuff to move. So some of them will be electrically gated. So you have to have a change in electrical charge. Some of them are chemically gated, so they have to bind with the right neurotransmitter or hormone. Some kind of signal would have to tell them to turn on. But for the most part, they're there, but they don't freely allow stuff to pass through until we turn them on. Okay, before we move any further, we're going to introduce a couple of terms that are going to be really relevant to talk about how the next stuff is going to happen. So the first thing that we want to talk about is the idea of polarization. And that's a state where there's a difference in electrical charge between the inside and the outside of a neuron. So if we have that phospholipid bilayer and we have our protein channels that are mostly closed, they don't allow stuff through, we can have a different set of molecules outside than we do inside. And if the ones that are outside tend to have a different charge than the ones that are inside, we can end up with this polarization where we have a charge difference across our membrane. So we can say that our membrane is polarized. We have that difference in charge stored across the membrane. And we're going to use these terms quite a bit. So the understanding of what they do is going to come. It might not happen immediately. The next of our terms is talking about voltage. And this is how we can talk about a specific measure of the difference in electrical charge between 2 points. Now, I always make it clear that voltage isn't sort of a single location. It's not an absolute value. A voltage is a comparison between two locations. So if we were looking at 2 points inside the cell membrane, there's no difference there because the ions that are inside and there's no difference between them. But if we have one sort of sample spot inside and one outside, then we're comparing across that membrane. We're looking at two different points. And what we end up happening is there's a difference in charge. We have that polarization across the membrane, and we can measure that in terms of voltage. So a higher voltage means that there's more of a difference across that membrane. And conveniently, storing those difference in charge across the membrane actually gives us a storage of potential energy. And that means that those ions would like to move around. They'd like to get across the cell membrane. Everything would like to equal out to zero, but they can't because the phospholipid bilayer is blocking them. So by preventing them from flowing the way that they want to, we have potential energy stored across that membrane, which means that if we opened up those channels, stuff would move and we could harvest that movement in terms of sort of a signal or an energy, which will become very relevant shortly. And we can talk about the resting potential as the difference in charge between the inside and the outside of a membrane in a neuron when that neuron's at rest. So when the cell is just sitting there, we tend to have a slight difference in charge between the inside and the outside. So we have a resting potential. When the cell's not doing anything, we have a stored potential across that membrane. The membrane is polarized, and we could measure the voltage of that difference in charge across the membrane. So lots of terms there that may be new, but we're going to keep using them and hopefully get familiar with them throughout this lecture and then the rest of the chapter. The last of the things that we want to talk about are ions. And ions are atoms that have lost or gained electrons. So electrons are negatively charged. So if you lose a negative charge, then you are left being more positive than you were before. If you gain a negative charge, then you've gotten more negative. So we can have positive and negative ions. We're going to look at a very small subset of those. For our positive ions, we typically look at sodium or Na+. So that just means that we have a sodium atom that has lost an electron, so it's net positive. We might also have potassium, which is K+. So that's a potassium molecule that has lost an electron, so it is net positive in charge. We can also have negative ions, so we have chlorine ions. That's the Cl-. It's picked up an extra electron. And then our last one is actually a weird one. You may be noting that it's A, and we don't have an element that's symbol is A. And that's because this is one of those weird things. I don't know if it's just psychology or if it's a bio thing too, but that's the symbol that they're using to indicate a broad group of organic anions. Basically, in order to simplify all the molecules that they point out, they introduced a new concept. So I don't know if it's actually simplifying anything, but A is a generic term for a bunch of other organic molecules that all conveniently have a negative charge. So we're hand waving what they are, but if you see A minuses floating around, we're just saying that they're genetic, organic, negatively charged things, which is what they want to do. Okay, so we have these ions, and we've kind of talked about this idea of having a charge, a difference in charges across our membrane, and how they want to be sort of back in balance. But I'm going to take a moment and talk just a little bit about why they would like to move, why the ones that are outside want to come inside, and why the ones that are inside want to go outside. What's creating that resting potential? What's driving this whole system that we're talking about? We're going to talk about two forces. And the first is a force of diffusion. And this is the tendency of ions to move to sort of from an area of high concentration to an area of lower concentration. You might have heard about diffusion in chemistry or physics. We talk about this term quite a lot other places. So this is the idea that we have a lot of these potassium molecules or ions here, and they don't all want to be together on one side of the membrane. They would like, ideally, to pass through the membrane, to pass through that gray bar, and spread out, take up all the available space, move to an area of low concentration. And this is one of those times that I have actually used PowerPoint animations as much as I dislike them, because movement is one of those things that does really well. This is also why for this chapter and this topic, I like to include videos on Moodle. Once we've gone through some of this stuff, I'll start sharing some videos that help go over it again. Because seeing them move instead of static images is a little bit more helpful. But that's diffusion. That's that idea of moving from an area of high concentration to low concentration. They want to spread out and fill the space evenly. Our next force is electrostatic pressure. And this is a force where ions are repelled from similarly charged and attracted to oppositely charged ions. That's a really fancy way of saying that the positive charges don't want to be next to other positive charges. The negative charges don't want to be next to other negative charges. The positives would like to be near negatives. Negatives would be like to be near positives. And if they had their way, they'd move or trade in such a way that we're net zero across that membrane. They'd like to have no charge difference if they could because of this electrostatic pressure. So if we're talking about this is how they want to move, but our membrane doesn't allow them to move freely unless we open up those channels, then we're talking about that idea of resting potential where they're stuck in places that are storing energy. They're stuck in this high energy position and they want to move across the membrane but can't. So how do we actually set up that situation? How do we create that high energy state where they're stored somewhere that they want to move away from? And that's where we introduce the last of our key components here. And this is the sodium potassium pump. And so again, the name gives us a little bit of a hint as to what it does. So it's going to move sodium and potassium ions, and it's going to act like a pump. So in this case, the pump is going to use energy from the cell, so it's going to burn ATP in order to move things sort of against the concentration, against the gradient that they want to follow. And so if you think of like a pump to fill a bicycle tire, so you can move it up and down. So when you move it up, it's going to sort of bring in air, and then when you push it down, it's going to force the air down a tube. Same basic principle. So our pump is going to have two basic motions, at least that we care about. We kind of hand wave the specifics here. But when the pump raises, it's going to move a bunch of sodium ions, those Na pluses, it's going to move them outside. So it's going to dump 3 sodium ions into that extracellular fluid when it moves up. Then when that same pump comes back down, it's going to push or sort of bring with it 2 of our potassium ions. Those are the K pluses. So every time this pump operates, every cycle, it's going to push out 3 sodium ions and pull in 2 potassium ions. Now you might be saying, hold on, if we're setting up a difference in charges, why are we only talking about positive ions? And that's because we've established this idea of that potential across the membrane of that polarization being a difference in charges. So what we're ending up with is 3 positive charges go out and only 2 come in, every time that pump cycles, we're ending up with net 1 extra positive charge outside and 1 less positive charge inside. So we're setting up a situation where the inside is going to be a little bit more negative compared to the outside. That's why we talked about voltage being measured as a comparison across something, comparing those 2 points. So we're saying that the outside is more positive and the inside is more negative. So we can talk about our resting potential, that setup where they're separated and there's more sodium outside and more potassium inside. We're sort of net negative on the inside as compared to outside the cell. So even though we're only focusing on positives, because we care about relative charge, we're saying that the inside is more negative, or at least less positive than outside. So when we start talking about actual numbers, we'll talk about the voltage across that cell as being a negative charge. And then that brings us back to our ion channels. So that's going to help us transition into actually moving into the next steps. So those ion channels, this is the definition for them. They're gated pores. As we said, they're proteins. They're basically tunnels through that membrane. And when we activate them, we can open those gates and we can allow the sodium ions that are stuck outside to come inside, or we can allow the potassium ions that are inside to go outside. And again, they can be chemically gated or electrically gated. So we can turn them on using neurotransmitters or hormones if it's chemical, or we can open them up using electrical signals. And once we open them, once that signal has been delivered, they allow that resting potential, that stored energy across the membrane to start moving. And that's where we're going to pick up in the next class. We're going to see what happens when we let things start moving again, and how we can sort of harness that movement and use it to transmit a signal. And again, we'll go over sort of the last part of this before we start again next time, just because this is a lot of information, lots of moving parts, and I want to make sure we're all on the same page. Yeah, that will be it for today.